Fantastic. Thank you so much. Really appreciate the organizers for their generosity in coordinating this teleconference. So today I really wanted to share a little bit about the work that we do here at RoosterBio in developing commercially relevant processes for mesenchymal cell manufacturing. So our goal as a company is to fuel the rapid commercialization of regenerative cures, and we do this by providing standardized cellular starting materials that could help companies who are developing MSC-based therapeutics to get into the clinic faster than ever before. So as many of you know here that MSCs serve as the workhorse for many regenerative medicine products because of their regenerative and immunomodulatory capabilities. MSCs are widely used today as a cell therapy for a variety of different indications such as stroke, heart attack, and graft-versus-host disease. Additionally, MSCs can also differentiate into bone, cartilage, and fat cells, and they've been used for a variety of tissue engineering and bioprinting applications. Now, the rise of gene therapy has also led to an increased interest in using MSCs as a cellular vehicle for delivering genes or targeting cancer. And MSCs are also known for their paracrine effects. And so recently you see a lot of interest in looking at the extracellular vesicles and secretomes of MSCs for the potential use for various indications as well. Today, there are over 950 clinical trials and more than 10,000 patients have been treated with MSCs. With a proven safety record, this really makes them a very attractive candidate for cell therapy. Now, regardless of the application, a critical bottleneck that is faced in the industry today is that cells are expensive and time-consuming to produce. This is a very typical scenario that we see with a lot of the people that we work with in the space. If you're starting with a competitor product with only 1 million cells, it will take you many, many days or even weeks and consuming many, many bottles of media before you can even get enough cellular materials to start your experiments. So as you can see here, the biggest enemy in any cell manufacturing process is the cost of media. So with this in mind, RoosterBio's first product innovation was to develop a bioprocess media that minimizes the use of media for MSC production. Now, some of you there might be thinking, wait a minute, Maya, this doesn't make sense for RoosterBio. Why is the company not trying to make more money by selling us more media? Well, our goal here was really to develop a more efficient process that makes sense for the industry as a whole, and to minimize the amount of waste that we're producing by consuming more media than we really need. So with one bottle of media and one bottle of cells, we want to get you as quickly as possible to the target cell numbers that you need to run your experiments. And our goal is to simplify your workflow, minimize your cost, and reduce the cell culturing time so that you can focus on your end application. So when I mention the word scalability to a lot of my academic friends, they usually give me a blank face and don't really know what I'm talking about. Okay, I can make this bad joke because I used to be an academic, but in all seriousness, most people underestimate the amount of work that needs to go into developing scalable processes for cell manufacturing. So every time I work with a new group, I'll ask them I'll start by asking them to think about where they want to go, right? What is your end goal? And then slowly kind of work backwards to to develop and determine what kind of processes they would need depending on what stage of the development program they're at. And this is really where we at RoosterBio share our expertise to provide the best practices in MSC manufacturing to support their programs. And this slide shows some of the standard 2D processes that we typically recommend our customers using our cells and media system, depending on what stage and what scale they hope to achieve. As an example, at the R&D stage, if you're trying to obtain 200 million cells, we would start with a 1 million cell vial, expand it into a T225 over 2 passages, and you'll get anywhere around 150 to 250 million cells. And then as you move along to your phase 1 trial, you may need as many as 3 billion cells. And here you'll start with a 20 million cell vial, and this is our standard cGMP cell vial, expanded in CS10s over 2 passages, and you'll get anywhere between 3 to 5 billion cells. Now, as you increase in your cell demand, basically what you're doing here, you're scaling out the process, right? And you're increasing the number of cell stacks that you'll be using. And as you can see here, at the 6 to 10 billion cell level, you will need to process 40 CS10s simultaneously. Now, I don't know how many of you out there have witnessed or have actually been in the lab processing 40 CS10s simultaneously, but it is definitely not a small feat, and it requires a team of people that is well orchestrated processes in order to process this quickly and efficiently so we don't compromise the quality of the cells. So you know where I'm going with this in terms of going towards bioreactors. But before jumping into the work that we do in bioreactors, I wanted to show you this slide on how the evolution of manufacturing platform is driving the economics of stem cell cost. So we're all aware that at the T-flask level, it's very much limited by the lot sizes, right? Anything beyond 1 billion lot size is going to be very challenging to achieve even with automation. So this is where we move along to the multi-layer vessels, and here you can easily generate billions of cells, but anything beyond tens of billions of cells is also going to be very labor intensive and may require automation. So we move along to the bioreactor scale, and here this is where you can generate hundreds of billions of cells with ease. And as you move along in the different technology platforms, you'll notice that the cost of producing these cells will also decrease accordingly. Now, there's a couple of publications that we have published around this work, and I don't want to go in too much detail, but I do want to reference these publications to you. And the first publication was published by John Rowley and our group in Bioprocess International in 2012. Subsequently, UCL group has also published in 2014 in Biotech and BioEng, and the most recent publication is in Frontiers in Medicine by our group led by Tim Olson in 2018. I also want to bring your attention to our blog, and in this blog, we actually highlight how to build a multi-year process development program talking about lot sizes and technology platforms that you should select based on the lot size. So definitely refer to this wonderful resource that is free and available for you for your reference. So today, I want to talk a little bit more about this process that we've developed in the PBS bioreactor systems. And this is a fed batch process where MSCs are seated in microcarriers and into these vertical wheel suspension bioreactors. And here we are using our Rooster nourished xeno-free standard culture media. And on day 3, we would add a bioreactor feed before we harvest on day 5. The typical densities that you obtain in a bioreactor culture is anywhere around 0.4 to 0.7 million cells per milliliters. Sorry. And here, this slide shows you a typical yields that you get at various scales in the bioreactor systems. At the 3-liter scale, you can get anywhere between 1 to 2 billion cells. And at the 50-liter scale, you could get as many as 30 billion cells. Now, one of the most significant step change in going into bioreactors is the increased media productivity. As you can see from this graph, our RoosterBio media is superior in terms of media productivity compared to our competitors' media. And this is because we have designed our media to minimize the use of media and therefore maximizing the yields or the output, right? So there's already a benefit in using the RoosterBio media systems, but as you culture this in a bioreactor, you can see that you have an additional increased media productivity, and our data here shows that you have almost a 3-fold increase in media productivity. Now we've validated the yields that we can get from these bioreactor runs, and we have conducted multiple runs performed at different times, and this data here shows the 3 independent runs from a 3-liter bioreactors using the same donor, and you can see that we're able to obtain consistent yields and performance from each run. Here we further demonstrate that we're able to get a very robust scale-up across different donors from as little as a 0.1 liter bioreactor all the way up to a 50-liter bioreactor. And in fact, you can see better performance. You see a faster growth in the larger bioreactors as compared to the smaller systems. We've also demonstrated there's good comparability of the MSC characteristics between the bioreactor cultures as what you would observe in the 2D cultures. And here, this data shows the tri-lineage differentiation and the cell surface marker expression for 3 different donors. And this slide here shows the IDO activity and the angiogenic cytokine secretion profile, again, for 3 different donors. And you can see that the profiles are very consistent from what we obtained to the 2D cultures. I also want to mention that this IDO and angiogenic cytokine profile that we're showing here is the standard panel that we perform for all of our MSCs as part of a characterization panel. So some of the key takeaway from this study is that when we culture MSCs in bioreactors, we immediately gain an increased media productivity. We see from our data that we get almost 3-fold increase in media productivity. We've demonstrated that we get very good consistency in terms of the performance across different scales from 0.1 all the way to a 50 liter. And we also show good comparability of the MSC characteristics with what we have observed in standard 2D cultures. So building upon this really strong process foundation, we can now further extend the MSC manufacturing to produce extracellular vesicles, EVs. And using the same manufacturing process for MSC production, we're now extending this culture 2 more days and switching to EV collection media to harvest the EV. Here we're using our RoosterBio low particulate EV collection media called Rooster Collect EV. And after you collect, then you can do downstream processing. So this graph here shows the MSC EV production in the bioreactor data. On the right side, you can see the cell counts at 3 different bioreactor scales, a 0.1 liter, a 3 liter, and a 15 liter bioreactor. And on the right side, you can see that this is the EV production measured by the particle concentration per milliliters of media volume use. And from this data, you can see that in the bioreactor, you actually get more higher particle concentration as compared to your 2D cultures. Excuse me. This slide is really a summary of the previous one. And we're really looking at the data from the perspective of EV productivity. So here, EV productivity is in terms of particle concentration per volume of media. In the 2D scale, you can see it's around 0.5 E9 particles per mil, but in the bioreactor, we can get more than double that. So it's around 2 E9 particles per mil. And we also see that EV production scales with volume, right? So basically, if you're using a bigger bioreactor, you get more EVs. At 15 liter, you can get as many as 10 trillion particles. In terms of characterizing the EVs, the particle size distribution in both the 2D and the bioreactor cultures are consistent with what we would expect of EVs. And our FTIR analysis also shows that the EV collected consists primarily of small RNAs. The RoosterBio MSC EV protein and tetraspanning content and localization are very much expected with what we would expect of EVs, as shown in our Western blot and immunofluorescent analysis. And we also perform a simple scratch assay to assess the bioactivity of the MSC-derived EVs and show that there is a complete closure of the wounds in the presence of the EVs. Now, all of this work we have done is really to support our customers to accelerate their programs and get them into the clinic faster and more efficiently. Here, I wanted to emphasize the focus on speed and accessibility. Our goal is to provide our customers with the tools and processes to help them develop the quickest path in any MSC-based therapeutics. We know that the road to develop any therapeutics is a long one and potentially with many unknowns ahead of us. But our goal is really to streamline the multiple stages of research, development, and clinical translation so that you can get into the clinic in a much shorter time. And our vision is to be the Intel microchip for every MSC-based regenerative medicine product so that product developers can focus on developing their cell therapy by leveraging off-the-shelf products that we develop to support their programs. In closing, I just want to mention that in November 2018, RoosterBio launched our first clinically relevant GMP starting materials for allogeneic hMSC-based manufacturing. We have master files with the US FDA for both our cells and media, and we're currently in the process of filing for JP and EU compliance. Since the launch, we already have several customers who are using our product and have referenced our US FDA master files. If you have further questions about this or any of the topics that I've discussed in this presentation today, feel free to reach out. I also provided here some resources. If you'd like to download a copy of these slides, you can go to info.roosterbio.com/BPI Asia 2020. If you'd like to reach out to me, you can email me. My email address is below, and or you could also reach out to me via LinkedIn as well. So thank you very much.